بسم الله الرحمن الرحيم. السلام عليكم طلبتنا الأعزاء. اليوم راح نبدي بأول محاضرة من الهيستوكيمستري. Histochemistry is composed of two words, histo and chemistry. This means the chemistry of tissues. It is one of the most widely used techniques. It helps researchers and scientists to localize or visualize cellular components of the tissue and other living structures by using chemical, physical, or biological techniques. The methodology of histochemistry, it consists of the first step, which is fixation of the tissue. This means when you take a biopsy of any part of the body or the oral cavity, you have to put this biopsy in certain fixative solution in order to preserve this tissue from being destroyed. So you can be able to examine this tissue later on. Also, it consists of a demonstration of biochemical components such as nucleic acids, proteins, sugars, lipids, enzymes, pigments, inorganic substances by means of chemical reactions. The technique uses different stains and indicators. This react with the cellular components or the targets parts of the cell or tissue we are looking for. This will develop tiny colored structures which could be later on easily observed or examined under the ordinary light microscope. The staining procedures and more specialized histochemistry and immunohistochemistry, they are performed to obtain specific types of information about bone cells, bone, cartilage, extracellular matrices, and bone formation or mineralization. And all these aspects cannot be usually obtained with one staining procedure or localization method. Sometimes you need combination of two staining methods. The staining techniques. The goal of the histochemistry is to provide color, as you mentioned, and contrast to the macroscopic images. So you can see the tissue under the microscope and examine all the parts you need. The field uses disparate techniques to accomplish the specific labeling of biological structures. You have to label the structure or the target you are going to examine inside the tissue. Histochemists pioneered the use of what? These small molecule cellular stains, labeled molecules such as what? Such as the antibodies, and enzyme-mediated detection, and signal amplification. These techniques are used in order to be able to examine certain structures or molecules inside the tissue you are examining. Histoenzymology or enzyme histochemistry. This technique is based on the capacity of some enzymes to keep their activity after tissue fixation. You know the fixative solution, usually we use formalin. It will cause a coagulation of certain proteins inside the tissue. This technique usually helps these enzymes to maintain their activity so that you can examine them later. These enzymes and the cell they are located in can be visualized after the conversion of soluble and colorless substrates into what? They will be changed into insoluble and colored products by the enzyme activity. Substrates are specific for the enzyme, and the products precipitate in the place where the enzyme is. There is a diversity of enzymes that can be detected with this method. Examples of these enzymes: peroxidase, phosphatases, dehydrogenases, acetylcholinesterase, and more others. These are the most common enzymes. So the site where the enzyme is present inside the tissue will be detected by producing a colorful substrate which is insoluble. Fixation and embedding of the tissues. It means you'll be fixation by the formalin and embedding inside the paraffin wax of the tissue may affect what? Affect enzyme activity. Embedding should be avoided because dehydration and high temperature may damage the enzyme and therefore its activity. That's why we use the enzyme histochemistry. which is mostly done on freezing or vibratom sections where no embedding is needed. So we can avoid the embedding or the side effects of these steps, fixation and embedding, by using the frozen sections or vibratom sections. And as I mentioned, there's no need for these steps. So we can maintain the activity of the enzyme, and later on we can examine it under the microscope. Now we are going to talk about the histochemistry of some oral tissues. Now again, this is another division for the histochemistry, which is based on the chemical reactions between cell components and stains. You know, this definition of this is, word or this term is variable, but just to illustrate what is the meaning of histochemistry. It depends on chemical reaction that occurs inside the tissue or inside or outside the cell, inside the extracellular matrix, between different molecules or different components of the cell or the extracellular structure. The end product of this reaction are permanent colored precipitates. The color will enable us to see the site we are examining. That can be viewed under the microscope. There are stains specific to each component of the cell based on the basic or acidic nature of the dye. Now, basic principles of histochemistry. It combines the method of histology and chemistry or biochemistry. This will reveal the biochemical composition of tissues and cells. This method means the hematoxin and eosin stain. This will uh, keep the normal distribution of the chemical without disruption. The application of this method, it will identify, quantify, and localize what? Localize the chemical substances, gene expression, biological structures and organelles, specific cell types. Also, it will clarify cell and tissue structure and morphology. Also, it will demarcate functional boundaries. There are some limitations of this method, as that we cannot use for real-time in vivo analysis for any tissue. It requires the removal and killing of the tissue. Uses in humans is limited to biopsy tissues. For looking at changes in tissues over time, each point in time requires a new tissue sample from a new animal. That's why when you do an experiment, we have to take several animals, and we can follow the changes that occurs in the tissue or the organelles of the cell or extracellular matrix uh, by following these changes throughout the healing. If it was a healing process, for example, wound healing, we can follow the changes that occur during the healing of this tissue, but not at the same sample. We have to take different samples over the over time, over throughout the healing periods. We can't depend on only one sample or one biopsy from one animal. That's why it cannot be applied to the human. Tissue preparation and histochemical analysis may alter specimen morphology, chemistry, or chemistry, depending on the methods and materials used. Now, the goal of histochemistry. Now, first, presentation of normal chemical distribution. The substance being analyzed must not diffuse away from its original site. For example, we are looking for some structure like an organelle inside the cytoplasm of the cell, or something you are looking for a certain structure outside the cell. We have to keep this structure in its original place so we can have a good idea about its function or shape, morphology. Second, the presentation of normal chemical composition. It means the procedure must not block or denature the reactive chemical groups being analyzed or change normally non-reactive groups into reactive groups. So this will maintain the chemical composition as it is without any change. Now, third, the specificity of the reaction. means the method we are going to use to stain the tissue it should be highly specific for the substance or chemical groups being analyzed to avoid false positive results. Detectability of the reaction product. The reaction product, as we mentioned, which should be colorful and insoluble, should be colored or electron scattering so that it can be visualized easily with the light or electron microscope. The insolubility of the reaction product, it means the reaction product should be insoluble, as we mentioned in the beginning, so that it remains in close proximity to the substance it marks. Structures and chemical composition of oral tissues. It includes epithelium and its derivatives, connective tissue, cells fibers, and ground substance. Now, the oral structures include, as we mentioned, the connective tissue, the epithelial lining, and associated glands. The significant chemical constituents of these tissues are proteoglycans, glycoproteins, mucins, and enzymes. Firstly, the connective tissue, what does it mean? It means that it is derived from the mesenchyme and consists of various types of cells, fibers that are embedded in amorphous semigel colloidal material, which is the ground substance. Now, the ground substance is a mixture of macromolecules. They show ability to interact with cells and fibers in the matrix. Thereby, they play an important role in adhesion and signaling events. The connective tissue ground substance is primarily composed of proteoglycans and glycoproteins. This is means that they are the main constituents of the ground substance. First is the proteoglycans, followed by the glycoproteins. Now we are going to have an idea about the proteoglycans. What are these materials? They are large molecules consisting of both cell surface associated and extracellular macromolecules. The proteoglycans are formed of a protein core, to which a large number of glycosaminoglycans chains composed of repeating disaccharides units. So it's a carbohydrate with a protein core. They may be unsulfated or sulfated. The unsulfated type as hyaluronic acid, or sulfated as chondroitin sulfate, keratin sulfate, and heparin sulfates. The heparin is highly sulfated glycosaminoglycan secreted by connective tissue mast cells. Numbers and types of glycosaminoglycans attached to the protein core and the proteoglycan molecules, as well as the polymeric state of proteoglycans, these determine the viscosity of the amorphous ground substance. Hyaluronic acid, it predominates in the loose connective tissues, and because of its high capacity to bind to water, is primarily responsible for transport and diffusion of metabolic substances across the tissue. Now, chondroitin sulfates predominates in the cartilage, proteoglycans, and are primarily responsible for supportive and somewhat plastic texture of this tissue. Chondroitin sulfates constitute only 1% of the total bone tissue, whereas only 0.5% is present in dentin. There are few proteoglycans which contain only less numerous glycosaminoglycans chains and are called non-aggregating proteoglycans. They include decorin, fibromodulin, which are seen in association with the collagen, perlecan in association with basal lamina and cartilage. The agreen is seen associated in association with basal lamina of a neuromuscular junction and glomerulus. Lastly, the syndican are the major proteoglycans of many organs as vasculature. Now, the other main component of the ground substance, firstly, we talked about the proteoglycans. Now, the glycoproteins, fewer associated carbohydrate moieties than those present in the proteoglycans. The carbohydrates are not present in the form of regular repeated units, as we mentioned in the proteoglycans. These are secreted into the connective tissue by epithelial cells and fibroblasts. Example, fibronectin, secreted by fibroblasts, smooth muscle cells, and various other cell types. The fibronectin is a multifunctional adhesive glycoprotein that exists in the extracellular matrix and body fluids. Laminin is secreted by the epithelial cells and present in all basement membranes. Chondronectin secreted by the chondrocytes. Osteonectin secreted by the osteoblast. These glycoproteins promote the attachment of the cell to their extracellular collagen matrices, and therefore they maintain normal cell morphology and control their cell function.